Bueno, buenas tardes. Tengo el agrado de presentar nuevamente a Nicholas Langlade del INRA. Thank you, Nicholas. He's going to present a second presentation. Esta mañana él hizo una presentación del proyecto Sunrise, de esta iniciativa público-privada. Focalizó en algunos traits como estrés hídrico, y ahora nos va a hablar sobre otro de los traits que focalizó el proyecto, que es el trait de senescencia. Nicolás fue un impulsor de todo lo que es la parte de fenotipificación, utilizando plataformas de fenotipado automático, y un impulsor de la integración de todo lo que es las aproximaciones genómicas, fisiológicas, biología molecular y fenotipificación para abordar traits complejos. O sea, sin más y para no consumir tiempo, le doy la palabra a Nicolás. Thank you.
Thank you very much, Ruth. Thank you to ASAGIR for this invitation today. So I will present you an ongoing project, so we don't have all the results. It's not finished at all, but it was very interesting to me to present it and emphasize the collaboration we have with INTA Castellar on understanding the genetic basis of leaf senescence in sunflower. So you all know very well sunflowers. I'm not going to introduce it. And senescence, I'm sure you're familiar with it as well. So here it's just a quick slide to introduce some context. Of course, leaf senescence is very important for the final yield because it has an impact on the photosynthesis the plant will manage during seed development and then fix the carbon. And also it will impact the transfer of this carbon from the leaves to the seeds. So at the end, studying leaf senescence is very important in general for the yield of sunflower, and especially in drought condition, since drought is inducing earlier senescence. And there is a lot of genetic variation for this trait, and this is an example of two interlines that have a fast senescence and a slow senescence example. So there is genetic background. The trait is very important. It is related to drought tolerance. So we were very interested in. And we did this in the frame of the Sunrise project. On this population that I presented this morning, it's an incomplete factorial design, so where we studied hybrids, and 36 male lines and 36 females, both public and private lines were included. And in this population, each parent is present at between 10 and 15 times, which means that actually we have a good estimation of the phenotypic value of each parent. And we have a multi-environmental trial, and so at the end we have between 274 and 454 hybrids evaluated in each field. These parents, they were sequenced. This is what I presented earlier. And based on the resequencing of these parents, we identified 14 million SNPs. And at the end, after all the filtering, etc., we work with 453,000 non-redundant SNPs that are both polymorphic in the male and in the female, and that have a minimal allele frequency of 10% in the population. So this was this population of hybrid, so it's a diversity panel of hybrids, was evaluated in general in 13 environments, and some environments with replication. So we have 18 replicates in totals. Over 3 years, and this represents 9,000 plots. And in fact, phenotyping the senescence on this trial, it's a huge effort. And so I counted it. It's approximately, it's a bit less than 1 million leaves that were counted during the whole phenotypic trial. So this was only possible because this is a public-private partnership with many private investment. And all this phenotyping leaf senescence was done by our partners, COSAD, Maïsadour, Soltis with his Limegrin and Euralis, RGT, Syngenta, and Biogema. And Terrinovia, that is the Technical Institute. So there were summer workers, students, counting leaves all over the places. So I'm really grateful to them. And this enormous effort was very successful because we were able then to measure this leaf senescence. So how did we measure it? Basically, of course, we transform the calendar date into degree day time. Thermal time, and we measure the senescence as the ratio of senescent leaves over the total number of leaves, okay? And we filter the data to keep only plots where we had the data on at least 3 plants, because usually we do 4 plants. So we had, we kept the data with 3 or 4 plants and with 3 dates. So we measured that 3 times after flowering. And we kept these genotypes, and in total, this is what I mentioned earlier, we have between 274 and 454 hybrids measured in the 6 environments over the 13. Okay, so this is our phenotypic data. And when we, just an example of how it looks, if we take just 2 genotypes, one with a slow senescence here with the 3 days, you see the replicates, and here with a fast senescence. So through time it's increasing because it's a ratio of senescent leaves. And this, what we keep is the slope of this model, and this is going to be our phenotype. This is what we call senescence. Okay. And now because we had this population, we could study the genetic control of this trait. And for this, we used the MLMM model, GWAS model. So Juan presented it earlier. So this is basically a way to scan the genome for markers here that are associated to the slope. Okay. And you take the best marker, and then you put it in the next model as a covariate, and you scan the genome until there is no more polygenic variants left. And then this gives you, after n steps, according to, well, Juan found, for instance, 21 SNPs. Usually we find much less, so I think this is very good for you. And then this gives a set of what we call between us suggested SNPs, because we are very careful with Brigitte. And then there is a step of EBIC that allows you to select the most reliable ones. So this EBIC step is working usually with flowering time or very irritable traits. When you work with this kind of traits, more complex traits, it's, usually you don't get any robust associated SNPs, and you keep working with these suggested SNPs. So this is what we've done. And we did also different GWAS models, so having an additive effect of the marker. You can introduce an additive effect and a dominance effect of the markers, because the way we selected the SNPs, we had the alternative allele in both the male and the parents. So we can actually estimate the dominance of the allele. We can estimate also the female and the male value of the allele. If the allele comes from the female or if it comes from the male, we estimate different values. We estimate this in a model with the interaction. So it's 4 different genetic models, and they give slightly different results. So, so in total, over the 6 environments, we found 266 non-redundant SNPs associated to leaf senescence. And when you recover the redundancy, it's about 2,000 SNPs. So now the question is, are these SNPs all located at the same places, etc.? So this is what we've done. So we have our 2,000 SNPs, and we develop a rule to define what is a QTL. And so if we have 2 environments, basically the rule is based that if we find SNPs that are co-localizing in 2 environments, we start to think it's robust and it should be considered. So here is an example. We have the chromosome, the p-values of the two SNPs, and here another environment, the p-value of this SNP. And if the space between two SNPs is less than 2.5 megabase, so it's quite large, then we can call all this region as a QTL, and we define the most likely position of the QTL as the lowest p-value here. So with this rule, we found actually 17 QTLs. None of the QTL is found associated in all the environments, but still there is one QTL in 5 environments over 6. So I think this is a very likely. And then 2 in 4 environments, 4 in 3 environments, and 10 QTLs are found in 2 environments. And when, in collaboration with Paola Fernandez and Sebastian Motion at INTA Castelar, they are long-time specialists of leaf senescence, so we collaborated, and they identified already a lot of candidate genes among the NAC transcription factor family. And so we looked if NAC transcription factors were also colocalizing in our QTL, and this was the case for 2 of them. And this transcription factor, they were described in many species to be associated with leaf senescence, so it's very likely that between these 2 QTL, one found in 4 environments and the other found in 2 environments, the NAC gene that are colocalizing might be the causal gene. So this is very encouraging, and so here we are now, our collaborators at INTA are doing the validation of this NAC gene in transgenic Arabidopsis, so the sunflower genes, so we can validate that this QTL found in sunflower in Europe in independent experiments are the same that the one that were identified in Argentinian work. So it's a very great science when it works all over the world, and we are very happy to collaborate on this. So now, what we can do also on a genetic point of view is not only detect the genomic regions that control the leaf senescence, but also try to predict if other hybrids will have a slow or fast senescence. And this can be done using genomic prediction. And so we did it, so the principle of genomic prediction here as a first step is to train the model. So it's a statistical model that you train on a data set here with both phenotypes and genotypes. And then you learn a coefficient for every SNP in your, so there were 453,000 SNPs, so you estimate a value for the effect of every SNP in your model, and this to be able to be the closest prediction in your training population. So this can be done here in our hybrid population on the orange hybrids here that were phenotyped also. And so we train the model with this here, and after we can check in the test population if, predict the test population with this model. So here the test population, in our case, it's the white part of this graph, where the hybrids that were not phenotyped. So we were able to estimate their value based only on their genotype. And then we have an estimated slope of senescence for all the population. So now how do we check that these predictions are correct? So what we did an average slope for our training set, and then we train the model and we estimate the average senescence of each hybrid. We also are able to do it in every environment, so we predict in this in every on the 6 environments, and then we have the variability. Here it's a principle in general. So we predict the genomic selection model, okay? and we have average senescence. Then we can grow the hybrids in new environments. We measure the senescence, and we check if the prediction matches the observation. And then it's a correlation on the ranking. And here we have, if we have a good correlation, like here at 1, or we can have a bad correlation, and we see if our model is correct. So we couldn't experiment, of course, all the population. Doesn't make sense. So we chose 12 hybrids that were not observed, but had fast senescence, and 12 not observed hybrids that had slow senescence. But in addition to not to take too much risk, we also checked if these hybrids, they were always senescent in the 6 environments when we predicted them, and this is what we call the stability index here. So here there are hybrids that are fairly unstable across the 6 environments, and here hybrids stable across the 6 environments. Here in the green they have slow senescence, and in red they have fast senescence. And so we had 24 hybrids that could be used to check if our model was correct. And when we did it in 4 environments in 2016, in 3 of them the correlation was really good. This is this correlation value of 0.7 is very acceptable for this kind of genomic prediction. In 1 environment in the north of France here, the prediction was really poor. In fact, there was a flood. It was very dramatic. The field was completely flooded at the beginning of the experiment. Somehow, of course, we kept only the plots that were supposed to be okay, but somehow it interfered, and the predictions are not good in this environment. So still, we have 3 environments in normal conditions that are very correlated. So we think the genomic prediction model for leaf senescence worked really well. And so that this approach can be used in the future to predict senescence and help breeding. But genomic prediction, it works well with related genetic material. Here, our population of hybrids are very much related because each parent were evaluated at least between 10 and 15 times. So each time we try to predict an offspring of this parent, it's going to work really well. But then for breeding and for more general purpose, you need to be able to train new models, etc. And you don't want really to measure 1 million leaves every year, okay? So you need easier way to phenotype leaf senescence anyway. So this is why we decided to develop new tools to estimate the leaf senescence. So this is done. This is done in the frame of the Sunrise project, and a new project, a public-private project, also from an association that is a bit similar to ASAGIR, but only for breeding, which is named Promosol. And in the frames of these 2 projects, we developed three strategies to phenotype leaf senescence. So the first one is using Hellaphen phenotyping platform. So the Hellaphen phenotyping platform, it's an outdoor platform like that, where we can grow 1,300 plants in pots outside, and there is a robot that is autonomous that can go around and weigh the plant and water the plant and scan the plant. And so here is the view of the robot with a scale, light barriers here in white bars, and some cameras. So it's oh. Do you think there is a mouse? Ah, yeah, I think it's okay. while the sound here is... Okay, so here you can see a video of the robot that we've done. It's as you say, it's takes the plant on the robot. It's waiting it and it's watering automatically. And so it's also with this light barrier, it's automatically acquiring a silhouette of the plant. So here it's examples of two plants. One in dry condition, early stage, in irrigated conditions, and we developed a software to segment the plant and do some automatic measurements. So here it's again it's a small GIF video of the plants growing. Within a week, so very quickly, just so you have an idea of the kind of image we can acquire automatically every day. So usually twice a day, the robot comes and takes these images, and we are in the process of developing the software to extract information related to senescence, overall senescence. But for the moment, it's not finished. Still, using this phenotyping platform, we did manual measurements. And so for this, we used a DualX, which is similar to SPAD, to measure some anthocyanin and chlorophyll contents. And here are two sets of genotypes. So some genotypes from the hybrids predicted with the genomic prediction, and the red being the fast senescence and the green the slow senescence. And also we collaborated with Inta Castellar and Manfredi with these inbred lines that are contrasted for senescence. And we can see that actually this tool with the anthocyanin and chlorophyll, they discriminate well the behaviors of these genotypes from Inta and from the genomic prediction hybrids. So this is complementary to the Hellaphen platform and the robot, and will help the description of the physiological processes that differentiate between these two this different genetic material. Another tool that we're developing for phenotyping is the Phenomobile. So with the Phenomobile, it's here we want to phenotype senescence and traits in the field, and so it's here again. I have a little movie. Okay, so it's kind of a tractor on caterpillars that goes it has a head that goes over the plot and scans the plot. On it there are multispectral cameras, visible RGB cameras, and LIDAR 3D point cloud cameras. I thought it was... Okay, so you see that the robot is here and the head, and it's going to scan the plot like that. I think it's... Sorry, I don't know where it is. Okay, so we do. We received it at the INRA Toulouse. So this is the first one. It's a prototype, and it's in Toulouse since 2017. And it was a similar Phenomobile was developed on wheat. And here, this is a drone view of the robot that is scanning the sunflower plots. So using this, we can acquire this kind of image in a visible light. and then segment the part of green area of the plot automatically. And so again, we are collaborating with informaticians and image processing informaticians to develop the pipelines. From the LiDAR, it's a kind of a laser that scans the plot, and then you don't retrieve an image, you retrieve 3D point clouds. So it's a cloud of points in 3 dimensions. Each time the laser is encountering a leaf or anything, then you have a point. And this is the kind of thing that you retrieve. And if you move the 3D point clouds, you can actually distinguish the plants and the leaves here. And so, so again, there is we just acquired last year these images, and we will try to extract some leaf area and angle of organs from this 3D point clouds to measure light interception and leaf area index, this kind of measurements that relate to leaf senescence eventually. And the last thing, still in the field, but that is more advanced, is the use of drones. We have 2 kinds of drones at the INRA in Toulouse, and the hexacopter here you see is widely used. So we can acquire with it RGB visible images that are used to count plants at the early stages. But what we developed for the green area index and leaf senescence is from multispectral images. So here it's a set of 6 multispectral images that are about capturing about the same plot area in the field at different wavelengths. And from this, you can extract the NDVI signal for the plot that separates the green versus the soil, basically. And so you can have a green area index for the plot. So this was validated. And then from this green area index, because we did the measurements through time, we fitted a curve to this evolution of the green area index and get the slope, the terminal slope of this function, and compared it to the genomic prediction for leaf senescence of our hybrids, and the slope of the green area index estimated using the drone images. And this is what you see here, this correlation with the fast senescing hybrids and the slow senescing hybrids. And there is a fairly good correlation between these two estimators of leaf senescence. One is predicted using the genome, the markers, genetic markers. The other one being measured using drone images and multispectral images acquired with the drone. So this illustrates the kind of tools that we want to develop to be able to screen rapidly leaf senescence and evolution in the field. And we hope that in the future we'll be able to identify some interesting genetic material, and then after collaborate with the physiologists to understand if this is, of course, cosmetic senescence or not, and if this photosynthesis is active, etc., and how much it impacts, on one hand, drought tolerance, because this is our main goal, and on the other hand, just generally the filling and the harvest index of the crop. Okay, so to conclude on this work in progress on leaf senescence, we identified 17 genomic regions that are associated to leaf senescence in our population. Within this region, there are 2 QTFs where we have a good candidate gene, and we actively collaborate on this to understand if they are the real causal genes. We developed a genomic prediction model that allowed us to predict correctly for related hybrids the leaf senescence. We then invested a lot of efforts in phenotyping, automated phenotyping of leaf senescence to be able to characterize more easily all the genetic populations, so including Hellaphen platform and with the DualX, and we are working also on the image analysis. The Phenomobile data was acquired, and we will start next year. At the end of this year, the analysis, the image analysis from this Phenomobile data, and from the drone data, we had we developed a model to estimate from the multispectral images and the NDVI the evolution of green area index, and this was related to the leaf senescence in our model. And finally, I want to thank the people involved in the analysis. So Fanny Bonafous was a PhD student in the team that did all the genetics, the GWAS, and the genomic prediction together with Brigitte Mangin. Nicolas Blanchet is in charge of the Hellaphen platform, and so he managed all the Hellaphen data that you've seen, and you've seen also him talking during the video. Philippe Burger and Rémi Marandel are the most involved into the drone and the Phenomobile, and you've seen them in the video as well. And Xiuliang Jin, who is the postdoc that did the image analysis, the multispectral image analysis. And of course, I want to thank all the people that counted senescent leaf during all these years. And I hope we managed to have high standards in our research to meet their involvement, and so to be able in the future to estimate leaf senescence without redoing this. And that's it. Thank you for your attention.
Yeah, thank you very much, Nicolas. I think we have time for a few questions. ¿Alguna pregunta, duda, comentario?
Nicolas, in the first part of your talk, you used a panel of hybrids, and you were using as a reference time from sowing in degree days. In that panel of hybrids, how stable or comparable were the dates of flowering of the different members of your panel?
Yeah, so of course we studied and we measured, we noted flowering time, of course. This is one of the basic phenotype, and it's about 7 and 10 days from the first one to the last one, so this kind of range.
Why not use antithesis as you use in some of the other?
Yeah, well, I guess this is a bad habit because we do, but we didn't think about it.
Half the duration of the stable leaf area index after flower, or it goes off the plateau.
Okay, yeah, maybe we see if we can check how it works with this.